Your practical will consist of 50 questions. And usually I don't give any extra credit for studying a group for the practical. But because I feel so rushed, if you study with somebody else, you can get 2 for the practical. Okay? And then, because you all have been so good, if you're here today, you can sign this sheet. I need a sheet. Anybody got a sheet? If you'll sign the sheet of paper for me, I'll tack on 3 more. Okay? So if you're here today, sign the sheet, I'll tack on 3 extra credit points. The other 2 you can get by study. That means I'm not going to give any extra credit on the practical. Okay? Good? Any? Lab. For the lab. For the practical. Practical 2. Does somebody have an extra sheet that I can borrow? Okay. Sign my book. Here you go. Oh, got it. Okay.
Here. Oh.
Just put your name on it.
Oh.
And then pass it around. If you want extra credit, put your name on that so I know you did it.
Where can I put my name?
Okay? For the practical, not the. That's different. Okay? So, you'll need to know these things. Test, the test setup, substrates, reagents, pH indicators, presence, absence indicators, results, the pH of the results, end products, enzymes, and how to evaluate the test. Okay? These are the things you're responsible for. Carbohydrate fermentation test, also known as a phenol red broth, triple sugar iron, SIM, sulfur motility, methyl red, Vogt Pascal, citrate, DNA hydrolysis, urease, gelatinase or gelatin hydrolysis, nitrate reduction, skim milk agar or casein hydrolysis, starch agar, and lipid hydrolysis or tributylin hydrolysis. Okay, they're all the same. On that, has everybody found the matrix? Does everybody know what I'm talking about when I say there's a matrix out there for you? Does everybody know what that means?
No.
Okay, some of y'all have been paying attention.
Oh yeah. Just make it
Oh
explain it again.
yeah. This is what the matrix looks like. It is on Blackboard for you. It has all the tests that are going to be on this particular. I have an echo happening here. I don't know what's going on. But it has all of the tests. There are 2 on here. The decarboxylase test and the deaminase test. Those will not be on this practical. We moved them because we didn't see a reason to start something before spring break and not be able to finish it. So we moved them to the 3rd practical. Okay? So sometimes we're continuing to work, and so we're doing some things in practical 3 that won't be on the exam. In this case, all of these things will be, and anything we haven't talked about, which are all these things, and they're coming, but they're coming in the 3rd practical. Of which the last 2, phenylalanine deaminase and lysine, ornithine, and arginine decarboxylases, they're on the matrix, but they won't be on this exam, this practical. Okay? The only things that'll be there are these guys. Okay? And I just noticed that I updated this, and so the one that's on Blackboard is not my current one. I will fix that today before I leave. It will be this current one will be there. Okay? All right. Any questions about what's going to be on the practical? Okay, so let's go through some sample questions. Although this is a review, it is not the questions that will be on the practical, but the questions that will be on the practical are going to be very similar. Okay? So, what do the initials SIM stand for? Sulfur motility, or sulfur reduction, or hydrogen sulfide, whatever. Okay? How does one inoculate an SIM medium? What does that mean?
A needle.
Straight needle. It's a motility medium. You go about 3/4 of the way down and pull it right back out. Okay? On the
Can you
rest
say
of
straight
these,
needle?
huh?
You can say straight needle.
Mm-hmm. Or inoculating needle, or just needle. It's all good. Okay, so describe the reaction in A. So now, because sometimes the images aren't clear, you'll have to listen to me if you're taking it with me in class. If you're taking it in an alternate format, I put enough description on it so that it's discernible. Okay? So what is the reaction for 2A? I have growth only at the site of inoculation, and there is no other indications. So S,
Negative.
I,
Negative.
M, most boring test ever. All right, that's Klebsiella pneumonia. Anybody else want to sign the paper? Hand it back that way. Okay. What about for B? I've got fuzzy here. I've got red there. S. I don't know what this means. Negative. I. M. Yeah. So this is S negative, I positive, M positive. Here I got black and I got red. S, I, M. Good. Why is this going to be hard?
Wait, what was it? I'm sorry, I'm trying to make sure I'm like.
S positive, I positive, and M positive.
I'm sorry.
That's okay.
I'm already here way back.
Yeah, I know. I wish we could do it in the other room, but I think there's a class in there. Let me go see if there's a class in there. Because we just be over there. No one reading this? Let's go. There's no class in there.